The structure of the antibody molecule illustrates the central puzzle of adaptive immunity. Antibodies are the secreted form of the B-cell antigen receptor, or BCR. Because they are produced in very large quantities in response to antigen, they can be studied by traditional biochemical techniques. Indeed, their structure was understood long before recombinant DNA technology made it possible to study the membrane-bound antigen receptors of lymphocytes. The startling feature that emerged from the biochemical studies was that an antibody molecule is composed of two distinct regions. One is a constant region that can take one of only 4 or 5 biochemically distinguishable forms. The other is a variable region that can take an apparently infinite variety of subtly different forms that allow it to bind specifically to an equally vast variety of different antigens. The antibody is depicted as a Y-shaped molecule with a constant region shown in blue and the variable region in red. The two variable regions, which are identical in any one antibody molecule, determine the antigen binding specificity of the antibody. The constant region determines how the antibody disposes of the pathogen once it is bound. Each antibody molecule has a two-fold axis of symmetry and is composed of 2 identical heavy chains and 2 identical light chains. Heavy and light chains both have variable and constant regions. The variable regions of a heavy and a light chain combine to form an antigen binding site so that both chains contribute to the antigen binding specificity of the antibody molecule. And that's the structure of the antibody molecule illustrates the central puzzle of adaptive immunity. Have a great day.